All right, welcome back, and we're going to now look at the next stage of endochondral ossification. And so on this slide, we can see both primary and secondary ossification. This is a little bit of a different color scheme. We still have the kind of pink-purple foundation color scheme, but it's very, very vibrant. And your slide is also going to be similar to this. It's one of the most vibrant slides that we have. It's actually one of my favorite slides, quite honestly, to kind of get our bearings of where we are again on this website. We're here, let me go to the top. We can see that we're still in chapter 5, cartilage and bone, and I scroll all the way back down again to this this bone here. And so it just is labeled as bone, but underneath they identify it as your epiphyseal plate with, again, an H&E stain. So let's go ahead and click on this one. And it's a lovely whole slide. We don't get to see the entire bone. And one of the reasons why is because the bone is bigger, like we're further in development. And so with that, the actual bone overall gets a lot larger, and it doesn't all fit on one of our nice, beautiful little slides that we have. And so we get to see maybe a part of it. We don't even see the entire rest of the epiphysis. We don't see the rest of the diaphysis even over here. So this whole view that we see is really just kind of one side of a long bone. So we're taking that fetal pig joint slide where we got to see multiple bones, we got to see an entire bone, right? 2 epiphyses. Well, now this isn't like that. The bone's a lot bigger. You can actually see the parts of the bone like with your naked eye. So if you took the slide and put it on a white piece of paper, which I tell students to do, you get to see like this, you can actually see the parts. Be careful of also how your tissue is oriented on the slide. So if you look at your slide, and it's this orientation that you see here on the screen, flip your slide like upside down, okay? So that when you put it on your microscope, and as we know about inversion, it's gonna kind of put it into the right view, right? And so you don't look at this upside down. It doesn't really matter how you look at it, but most students, with the first time you're seeing your epiphyseal growth plate, you kind of want your epiphysis to be at the top. You want to kind of like north epiphysis and like the south to be your diaphysis, and to look at your growth plate kind of coming down. And so again, if your tissue looks like this on the slide, flip it. So when it gets to the microscope, it actually does have this orientation. Over here on the side, again, we get to see a nice little guide that is showing you the different zones of your growth of their endochondral ossification. It's nice because you can actually click on these zones here and it will kind of zoom you right in, which is fantastic. And then there, again, kind of a cheat sheet. We have some osteoclasts that they actually found in this, this gigantic area, right, of tissue development. They actually will pull you in. But again, at this low magnification, again, let's just get our bearings of where we are. We can see that this is the head, essentially, of the long bone. This is going to be the epiphysis. So this is all going to transition into, and it is a spongy bone, and it's going to fill in with red bone marrow. You can clearly see the band of the growth plate that is here. I think we get confused because you have one hot pink, bright pink red area, you got a purple band and then another purple band, and it's like, what am I looking at? And so at the bird's eye view and kind of a 4-power view, you might not be able to understand what you're seeing until you get up in there a little bit, but that's okay. I just want you to get your bearings first, and then we kind of start creeping downward, kind of through that metaphysis into the diaphysis area, and you get to see like even more development. So this is thickening on the sides, and that would be happening over here too if we had it. You can see that all of this like hot pink, deep pink color here is starting to thicken as well. So those spicule to the trabeculate are getting more dense. You're going to start having some remodeling of the spongy bone at the peripheral surface, which will become the sides of the cylinder, like the actual diaphysis surface, that will be remodeled essentially into osteons. So we're that one step beyond. So you have to think of your fetal pig development slide as being only primary. This is the next step. We're getting into primary, that's still continuing, but also secondary ossification. You can also see that there's a color pattern, that there's this bright kind of pink reddish color here. Well, guess what? You also can see that down here, which tells us that this is all has gone from calcifying cartilage into the ossification process. So this is being essentially ossified into actual bone material. You can look at all these lovely trabeculae in there too, and all this kind of purple white background is going to be all spongy, red bone marrow. And so spongy bone and red bone marrow, and then that color also is down here, which tells us, right, that that is bone too. The purple that you see, and again along that growth plate area, tells us that that's cartilage, right? Because the growth plate is going to remain cartilage until this organism is an adult, and then it will actually ossify and seal, and there will be no more active growth plate that exists. So I'm going to pull this down just so you have a little bit of a guide that is here. So where do I want to go first? What I think I'm going to do is jump right into the growth plate, because this is kind of why we're looking at this particular slide, but I'm going to go north and I will go south, but I want to do the zones again with you because this actually is difficult for some students. Let me drop this down. So the first area of our zones of our growth plate is resting, right? So it's our resting zone. And when you look at this section, I want you to go to the surface, the actual outer surface of the bone that you see here, and then kind of look into the actual growth plate. Because what you can see is that there's this resting cartilage here. It's like it's lighter. It looks like hyaline cartilage. There's more matrix. The cells are a little bit more uniform in their distribution. And then as you travel along, it like, where does it go? Where does it go? It like disappears. And so if you start like right here in the middle, and we kind of zoom in and zoom in and zoom in, and then I have, the question that comes up like, where's my resting? Like, is it this? Is it here? Like, where's the resting? And honestly, it is this little bit of light purple that is your resting cartilage. That is literally how small it is. Why? Because your bone has so much development. This is all ossifying up here. So this is all bony material at this point. Anything that's purple is going to be cartilage. So that's why I say, okay, you want to see a little bit more resting at your epiphyseal growth plate. Sorry, I'm going to drag you all the way back over here. And this is what you'd have to do on your microscope. Go, kind of slide, and like, oh, there we are. So we're really at that side area, and you can again see it's almost pink, right? It has this pink to purple color, kind of our true typical resting cartilage that would be out here to the periphery. And then, oh, look how gorgeous this is. Look at those columns. So just beautiful columns, these parallel columns of our chondrocytes. And so this is really, really kind of indicative of our proliferative zone, right? So the fetal pig slide definitely showed you this, but it's so much more exacerbated. Like here, it's so much more obvious here within the growth plate. So even if we travel down, we'll see the same thing. Like the cells are really close together. They're stacked one on each other. Look at all the nuclei. They're just like, like all the way down. You really almost don't even see lacunae, and they're there, but they're just so compacted. And then the kind of comparison, right? So if we even kind of get more in the middle, let me kind of just shoot on over, like you almost don't even see resting here, right? It's like, where did it even go? So sometimes I tell students it doesn't matter, not unless you really want to see the whole kind of column of development. So let me kind of come over here. You can see resting cartilage. Oh my goodness, look at the proliferative zone. And then right about there, it's like, oh, we get a little bit of a textural change. You get a pattern change. And then that's where we're going to hit the hypertrophy zone. So the zone of hypertrophy. Look at the cells themselves. In this case, the cells are actually getting bigger. Like the cells get bigger, the lacunae get bigger. In some cases, some of the slides, you won't actually see the cells. In some cases, you will. On this slide, look, you can actually see the nucleus. You can see the cell cytoplasm. It's just, they're huge. Everything is really, really big at this point. And then it's like, again, that lineage of that line, right, of change. And it's like, whoa, we're really changing into what we refer to as the calcification. So this is the zone of calcification, and we're going to start to see a color shift that happens here as well, right? And we also see blood vessels, we see bone marrow, we have just like a massive change happening right along that line. So if I zoom out, it's pretty obvious, right? Resting is here, proliferative, and like right about there, we get into hypertrophy and then the line of calcification. And then even as we drop down, like even if I put the pointer here, right? Like this to here, like this area is calcification. And then you get to see more of that brighter, deeper pink color, which really tells us that this is kind of calcifying cartilage, and then we're really starting to make bony material down here. Look at all these giant kind of trabecular structures. This is all like true bone. You might still get pieces and little bits of cartilage in there, that purple, but you really have big chunkier pieces of bony material. Lots of blood vessels come in here. We still got bone marrow. Even if we were to zoom in, right, we get in closer and closer and closer. You get to see these osteocytes. You can almost start to make out some of those lacunae that are there. The cells that are right along the border, those would be like osteoblasts that are there. So how is that different? And again, look where we are, okay? How is that different? Let me zoom out. Then what is up here? Well, these are going to be chondrocytes, right? So if we go back up here, I'm just curious. Let's see if we have any chondroblasts up in there. Yeah, we do. Like we have some chondroblasts. We don't really have lacunae that are around these guys, so they're still proliferating. But then up here, we can definitely see that there's white spaces around these cells. So those would be our chondrocytes. Let's just kind of pick a place in here. I'm just going to kind of pick a place and like, look at that. That is just amazing. Like lots of marrow. These again are all blood vessels, just a ton of blood vessels all over the place. And then just big chunks of bony material. If I go out to the border, and you can see I'm kind of traveling along here, I'm going out to the surface. It's going to start to thicken up. It's going to start to remodel more out here. You're going to start to see more osteon formation. Some of your slides, if I come down farther, I don't know if I can see that on these or not. Let me brighten my screen up a little bit. Sometimes it's like almost too bright. You can almost start to see that circular formation, right? You get to see osteons starting to form. And so this is kind of that remodeling area. Look at that. That's kind of cool, isn't it? But we're deeper down into that bone. So look at that one, right? You get to actually see that happening. That looks a little bit different. And look where we are. If I jump up here, right, we don't really quite see that as of yet. Let's jump back down, right? And we get to like see it. This is much easier on your slide. You're kind of like toggling back and forth, but you get the kind of general idea. So you can possibly see some of those osteons in development. Look at the color, right? It's just so vibrant. It's so obvious. Some other things here. Let's cheat. We'll just use our little guide, and we're going to try to find an osteoclast. And so let's zoom out a little bit. Okay. And then you can kind of identify them right away, can't you? It's like boom, 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 boom. And so this guy here is an osteoclast. And the reason that we know that, sometimes the cytoplasm actually stains a little bit more eosinophilic, which means it has more of a pinky orange color to it. But there's a border to the cell. It kind of like looks uniform, or it almost looks like all these nuclei are kind of like clustered together. So these cells are multinucleated. We have many, many nuclei within this cell. I don't see the Hauschipp's lacunae, although I wonder, if we can have a different view of it, if that kind of would be what we see there. Let's go to the second one. Let's see what that guy looks like. Oh, there we are again. Here's our lovely cell. That actually is a stack of red blood cells on each other, which is kind of cool. Let's go out again. Let's see what the third one looks like. Oh, that one's huge. It kind of has a little bit of a border, but it's kind of out here in space a little bit. But again, once you kind of see one and you get the pattern or the color of the cytoplasm, they're a lot easier to see. And again, remember that you don't get to see everything in a perfect view. You might get to nick an osteoclast, or you might get to see a whole one, like this guy. But again, like once you see one, you can't unsee it. So there's one, right? There's one. There's, again, another one over there, right? And so sometimes it's good to go, like what I've been doing, going out and in, right? There's one, and there's one. Why so many osteoclasts? Well, because we're remodeling, right? We are going from calcified cartilage to spicules to tuberculae to osteon formation. And so there's all of this building and tearing down that is happening on this slide, which is pretty great. let's head back out, and we could kind of zoom in here. Oh, maybe we could do that. And let's see if, let's see, that actually looks like muscle out there. But this would be like that periosteum that would be forming. That would be here. I get that question a lot of like, where's the medullary cavity? Well, let's just zoom out. Like, do you see any cavity? No, you don't. This will start hollowing out a little bit more. You'd have other membranes coming in. You still see a little bit of trabecular bone there, and it would start to fill in with fatty bone marrow. But we're still developing. We're quite not there yet, right? So let's take this color here, this kind of bright pink red color, a little bit of orangey tint, and match it up here. And so we are now looking at the epiphyses, right? And you note that this color here is a little bit different. It's not quite matching purple, but it is a little bit different. So we have this remnant of cartilage that will cover the articular surface, right? We want the ends of these long bones to retain some of that hyaline cartilage. So if we were to, let's see, let's pull this up, zoom in, let me pull it down. There we go. We would see, look, it's cartilage. It is retained cartilage, so it will stay that way and kind of retain as hyaline. If we kind of scroll down here, we start running right into kind of these trabecular regions. We get to see that, it does look like typical trabeculae. We get to see lots of marrow that is in here too. You would absolutely be able to see osteoclasts in here. The osteocytes are basically getting trapped inside of the matrix. You have osteoblasts at the surface. I just need to pull it. Actually, that looks like an osteoclast right there, doesn't it? It's like seek and find. It's the where's Waldo of cells. So that guy, if it actually focuses on, that would be like one of our osteoclasts. Again, lots of lovely bone marrow that is here. And again, if I zoom back out, it just has that pattern, right? It just has that trabecular pattern to it. I'm trying to think if there's anything else that we really want to see here. Another deeper dive would be getting into like identifying marrow cells and all of that. Again, you can see all the cartilage. You also can see, in some cases, like tendons and ligaments coming in and attaching. You could see muscle attaching as well, which is kind of cool. This is a bunch of fatty tissue up there. As we get further into the different slides, you could come in and say like, oh, like what's all this like good stuff over here to the side? And that's going to be muscle. So that's going to be like a bunch of muscle tissue. Should be striated. So let's see if we can see those striations. Oh, we can. And so you can see that these are all skeletal muscle cells that are right at the periphery of the bone, which makes sense, right? So we would have muscle right at the periphery or flanking our bone tissue that's being formed. Okay, other things that you can do on this slide, you can go ahead and kind of click these terms over here to the side. That's confusing. If you click that, it takes you here. You're like, what am I looking at? And so again, the resting zone would basically be right here. That's why I kind of took you to the side to kind of show you what resting is, and then you can kind of tease it out a little bit. And then of course you can kind of click into all of these, and pretty much what is right in the middle of the screen is what they're going to be showing you. Okay, so go ahead and play around with that if you would like to. I will see if there's any other slide that would pertain to you guys, and make sure you check out the other cartilage and bone videos as well if you need to. All right, thanks guys.